Welcome to all enrolled students. This FDP on Biotechnology, Engineering Life, focusing on harnessing biological systems for a sustainable future. Please note that there will be a test of every module, and a certificate will be given only on payment of $19.99/RS before the 20th of June, 2026. After that fee will be $2,999. This will be a foundation course. Advanced course fee will be 6,500 rupees, only one time.
Our strategic roadmap is divided into 3 phases. The foundational phase covers rDNA basics, safety, and literature review over 30 days. The discipline of biotechnology is an interdisciplinary fusion of natural science and engineering. Biotechnology is defined differently by various entities, but generally it's technology based on biology, particularly in agriculture, food science, and medicine. Practical applications span medical, agricultural, industrial, marine, and environmental sectors. The term biotechnology was coined by Carl Ehricke in 1919. He envisioned converting biological materials into raw materials for human use through engineering principles. Today, it represents the intersection of biological systems, living organisms, and advanced technologies to improve human welfare. Biotechnology is a fusion of natural sciences, including microbiology and genetics, and advanced engineering, which uses computational modeling, chemical engineering, and robotics for scaling production. The core pillars of biotechnology are genetic engineering and bioprocess engineering. Genetic engineering involves the direct manipulation of DNA sequences to change an organism's traits. This includes gene expression, cloning, and trait enhancement, essentially editing the blueprints of life. Bioprocess engineering focuses on mass production scale, using specialized containers called bioreactors to ensure sterile conditions for large-scale production of life-saving drugs like enzymes, antibiotics, and proteins. Biotechnology applications are often categorized by color-coded branches, providing a globally organized categorization of applications. Red biotechnology focuses on medical and health applications, such as developing recombinant insulin, personalized gene therapies, and rapid diagnostic kits. This has revolutionized the treatment of chronic diseases at the molecular level. Blue biotechnology explores marine and aquatic resources, focusing on discovering novel compounds for medicine, aquaculture improvements, and marine environmental protection. It leverages the untapped potential of our oceans to solve global challenges. Green biotechnology is centered on agriculture and food, aiming to improve crop yields and food quality. This includes developing pest-resistant crops and nutrient-enriched foods, crucial for global food security. White biotechnology, also known as industrial biotechnology, utilizes biological catalysts for industrial processes to reduce energy and waste. It focuses on eco-friendly biofuels and is vital for a greener industrial economy. Gray biotechnology is dedicated to environment and protection. It employs bioremediation, using microbes to clean oil spills and detoxify industrial waste. It is essential for restoring damaged habitats and maintaining biodiversity. This matrix summarizes the 4 main branches: medical red, agricultural green, industrial white, and environmental gray, along with their focus areas and real-world applications. Next, we will explore the essential techniques that form the molecular toolbox of modern scientists, allowing for manipulation of genetic material. This molecular toolkit includes DNA cloning, PCR, and electrophoresis. DNA cloning creates identical copies of gene segments. PCR amplifies tiny DNA segments into millions of copies. Electrophoresis separates DNA, RNA, or protein fragments by size and charge. Decoding the genome involves DNA sequencing to determine the exact order of nucleotides in a DNA molecule. The cost of sequencing a human genome has drastically fallen, from $100 million to under $1,000 in 2 decades. The chart illustrates the exponential reduction in the cost of genome sequencing, following a trend faster than Moore's law. This decline unlocks new opportunities in personalized medicine and research. Now let's trace the evolution of biotech and explore its legacy of discovery and breakthroughs. We see 4 major historical milestones: the coining of biotechnology in 1919, the discovery of the DNA double helix in 1953, the first recombinant DNA in 1973, and the completion of the Human Genome Project in 2003. As James Watson said, "We used to think our fate was in our stars. Now we know, in large measure, our fate is in our genes." We now move to the cutting edge of the field, exploring emerging horizons in biotechnology from 2024 to 2030. Revolutionary technologies driving the field include CRISPR-Cas9 for gene editing, mRNA vaccines for programmable medicine, and bioprinting for tissue and organ creation. Biotech is advancing with prime editing, a search and replace method that eliminates double-stranded DNA breaks, reducing errors and enabling the correction of a vast majority of human genetic disorders. Industrializing medicine through cell shuttles addresses the manufacturing bottleneck of cell therapies, replacing manual scaling with enclosed, automated bioreactor cassettes. This significantly cuts production time and operational overhead. Vascularized 3D bioprinting overcomes the challenge of tissue thickness by printing sacrificial networks that flesh out to create functional microvessels, critical for organs and tissues. Green biotech is focusing on climate-resilient crops. This includes polygenic modifications for stress control, fine-tuning stomatal kinetics for heat spikes, and halophilic engineering for survival in high-salinity soils. Rhizosphere engineering targets non-leguminous crops, such as maize and wheat, to associate with nitrogen-fixing microbes. This modification mitigates global nitrogen runoff and reduces the reliance on chemical fertilizers. Blue biotech is exploring extremophilic bioprospecting, which looks for organisms thriving in extreme conditions. This can yield stable enzymes, called extremozymes, for industrial use, and unique antimicrobial compounds. White biotech focuses on de novo catalyst design. We use computational modeling and machine learning to build catalytic active sites from scratch. This creates enzymes for xenobiotic degradation, such as breaking down PET plastics with high kinetic efficiency. Bioreactor engineering is crucial for carbon capture. This process involves industrial flue gas input, fermentation by acetogenic microbes, metabolic rerouting to produce acetyl-CoA nodes, and an output of sustainable aviation fuel and polymer. DNA archiving leverages the high density and thermodynamic stability of DNA for data storage. Barriers remain in reducing the synthesis, or write, and sequencing, or read, latency. This chart quantitatively compares the 4 core biotechnology branches: red, green, white, and blue biotech. They address global challenges, from personalized medicine to environmental protection. Looking ahead, future biotech horizons include precision proteomics for custom drug delivery, neural interfacing using organic semiconductors, and synthetic ecology to restore degraded biomes. Addressing biorisk requires strict biosecurity protocols. The dual-use dilemma demands careful consideration, while genetic safeguards prevent environmental survival and digital biosecurity screens DNA synthesis orders. Global bioethics emphasize the distinction between germline and somatic editing. Regulatory friction is a major challenge in approving modified organisms, and we must address the access divide to prevent inequity. Biomanufacturing is experiencing a shift in capital investment, moving from single drugs to long-term high-yield biological platforms. The scaling path from bench to commercial production is critical. Our 2030 timeline predicts major milestones. Between 2026 to 2028, we expect clinical readouts for in vivo prime editing and successful biofertilizers in corn crops. From 2029 to 2032, anticipate AI-designed plastic recycling enzymes and trials for tissue patches. Beyond 2033, we may see DNA storage data centers and approval for bioprinted transplant organs. To summarize, the core pillars include programmable nature, cross-disciplinary approaches, and global solutions. Biotechnology is systematically designed to solve global challenges. This presentation encourages a discussion on how global regulations should adapt for AI-designed synthetic organisms and the structural changes needed to scale cell shuttle therapies.